Think about the last time you scraped your knee, or look at how much a child grows in a single year. Your body is constantly repairing and building itself from the ground up, and it does this by making millions of new identical cells. This process of somatic or body cell division is called mitosis. The goal is straightforward. Take one parent cell and produce two perfect identical copies, preserving the exact same genetic instructions, a state known as 2N. Looking at this diagram, you can see the process is split into two distinct phases. First, we have karyokinesis, where the nucleus and the genetic material inside it divide. Only after that's finished does the cell move to cytokinesis, which is the physical splitting of the outer cell body itself. That first phase, karyokinesis, happens in 4 precise chronological steps. You can remember them with the acronym PMAT: prophase, metaphase, anaphase, and telophase. Because the DNA is the master instruction manual for the entire cell, it's highly fragile. The cell has to meticulously sort, package, and split this control center first, ensuring no genetic information is lost before it attempts to tear its physical structure in half. Looking at the top right of this illustration, we see the first step, prophase. Normally, a cell's DNA is a loose, tangled thread. To transport it safely, the cell condenses this material into tight, manageable packages. As they condense, they become short, thick, and visible as these distinct X-shaped chromosomes. While this happens, the nuclear membrane begins to dissolve, and structures called centrioles start migrating toward opposite ends or poles of the cell. Moving down to the next illustration, we hit metaphase. You can think of M for middle. The nuclear membrane is completely gone, and all the chromosomes migrate to the cell's equator, arranging themselves in a perfectly parallel, single-file line down the center. Notice these faint thread-like lines extending from the poles. These are spindle fibers, flexible protein structures that act like lassos, attaching directly to the center point, or centromere, of every single chromosome. This single-file formation is a strict physical prerequisite. By lining everything up dead center before the split, the cell guarantees that exactly 1/2 of the genetic blueprint